webinar of the National Early Detection and Rapid Response Framework webinar series. I am Kaylin Clements. I'm a social scientist for the U.S. Geological Survey, and I'm also the Partner Engagement Coordinator and one of the co-chairs for the National EDRR Framework, which is an initiative to strengthen interjurisdictional coordination and capacity to efficiently detect and respond to new invasive species across the nation. This series is a collaboration between the Association of Fish and Wildlife Agencies and the Department of the Interior to share information about the National EDRR Framework and discuss important topics with state agencies and other organizations involved in early detection and rapid response in invasive species. Carrie Wickstead, the Invasive Species Program Manager for AFWA, here with me today, is working closely with us on this series and is co-facilitating the webinar with me. In addition, we have more than a dozen scientists involved in the National EDRR Framework and who have expertise in invasive species and/or environmental DNA on the line who are helping facilitate today's webinar. Thank you all so much. It would be great to learn a little about where our audience, where you all are coming from today. We have a lot of people, so I won't do this one by one. If you'd like, please take a moment now to post in the chat your name, your title or job, maybe what organization you're affiliated with, and if you like any experience or interest related to eDNA, if you have. And then folks who are involved in the National EDRR frameworks, our facilitators, note takers, project leads, please do the same and maybe also mention what your tie is in the National EDRR framework. What project are you working on? If you're not seeing the chat, click the chat icon on the bottom of the screen. I think that's where you should find it and it'll pop up. So to give you some context around today's webinar, like I said, it's the third in the series. The first two webinars were last spring, and the first one included a foundation, sort of an introduction for understanding environmental DNA and its application for EDRR, as well as small group discussions on eDNA with the audience. If you missed it and you'd like to watch the presentations and main room discussion, please see the link at the bottom of the Wakelet, which Carrie, if you would put it in the chat, it would be a great help. And I'll explain what a Wakelet is in a second. The second webinar last spring focused on horizon scans and hotspot analysis and included discussions again with the audience in breakout rooms about how to make a public-facing online hotspot analysis tool most useful for on-the-ground applications. But today's webinar will begin with a presentation on a state-federal collaboration demonstrating application of eDNA in the Great Lakes Early Detection and Monitoring Program. And we have a guest speaker from the Michigan DNR with us today. I'll introduce in a moment. Then we'll hear about an effort supported through the National EDRR Framework to develop a molecular lab network for EDRR work. And throughout our presenters will touch on ways that the National EDRR Framework and eDNA tools in general can help address some of the barriers, concerns, and opportunities participants have shared in previous webinars. Then we'll go to breakout sessions to hear from your on-the-ground perspectives, or whatever perspective you bring on potential applications for eDNA. Like I said, our presenters have designed their talks to address some of the things we've heard from you. Thank you so much for everyone who provided feedback throughout the past year. Either when you registered, you answered some questions. There was a survey that went out over a year ago and at previous webinars. We've received an overwhelming response. We won't be able to cover everything that you want to know about, but there will be other resources to share and also future webinars that cover some of those things. One of those resources is the Wakelet that I mentioned. It's a web page that has links and PDFs that we refer to today and other materials like information about the National EDRR Framework and eventually the recording of this webinar. After our presentations, we'll have Q&A, so please feel free to post questions as they arise throughout the presentations. Presenters and other scientists on the call, please feel free to respond to the questions, but we'll also, like in the chat, or you can share links and resources. But we'll also have time for questions after each presentation, and we'll open it up and have presenters answer. We'll then disperse into breakout rooms for facilitated discussions on some discussion topics So please keep yourself muted during the presentations to avoid background noise, but when we're in breakout rooms or all together here, you are more than welcome to turn your video on and speak up if you have a question. So I'm going to just turn it over to our first presenters here and introduce them. Nick Fraunhower is our first presenter. He's a fish biologist working out of the U.S. Fish and Wildlife Service Region 3 office in Bloomington, Minnesota. He coordinates the region's eDNA and Great Lakes early detection and monitoring programs. He has worked in the aquatic invasive species realm for over a decade. We're really happy to have you here with us today, Nick. Thank you. And then also Dr. Lucas Nathan, who is the Aquatic Species and Regulatory Affairs Unit Manager for Michigan DNR Fisheries Division. As part of his role, he oversees the Fisheries Division's Aquatic Invasive Species Program. In addition to his work with Michigan DNR, he has 9 years of experience conducting genetics-based fisheries research, including 3 years of eDNA projects. So I think we have the right people here to teach us a little bit about their experience with eDNA. And with that, I'll turn it over to you, Nick.
Thank you, Kaylin. As Kaylin mentioned before, the first webinar, set the stage, built that foundational knowledge of eDNA. And Lucas and I will take the time today to talk about how that's applied in specific programs. I'll talk about the Great Lakes Early Detection and Monitoring Program, and then Lucas will take it over and talk not only about how it plugs in, how his state partnership plugs into the Great Lakes Basin program, but also how they are utilizing eDNA beyond that and within on special projects within the state. So with that, next slide. The Great Lakes Early Detection and Monitoring Program was established in 2013 with an initial framework completed in 2014. It's unique. It was the first of its kind. It has a large geographical footprint, involves 4 U.S. Fish and Wildlife Service field offices, 8 states, 2 provinces, and many other governmental and non-governmental entities. I want to stress it's an adaptive program. A few years back, we established a vision, mission, and objectives to provide clarity and common understanding for those involved with the program. And the reason I bring this up before I dive into it is they serve as guideposts for what we do and how we adjust, right? And so there's a lot of writing on here, but I want to emphasize that this is a collaboration and coordination with partners. And one of our objectives is to increase the probability of detection for aquatic invasives and species. Next slide. Given the large geographical footprint and the number of partners, as you can imagine, there are a lot of moving parts, a lot of information going into and flowing out of the program. And it can seem overwhelming, but I'm going to quickly step through it. I'm going to utilize this process visualization on the left to help kind of guide us through the program from beginning to end. Next slide. So it starts up here, and I affectionately call this the unicycle. So we're in the upper portion, and we start out with horizon scans. For the Fish and Wildlife Service, we have 2 fantastic people, Kate and Heidi, who head up these horizon scans in which they look at species from all over the world and what is most likely to impact the Great Lakes. From that, we use that information, next slide, and flow down into developing a watch list. We not only use those horizon scans, but we use information from other entities and partners. The governors, Great Lakes governors and premiers have their 10 most wanted species and other lists to feed into this list that we utilize for our program. And I have 2 species highlighted, big head carp and silver carp. Keep those in mind. In discussions with our state partners, they said these 2 species are their highest priority and the ones that keep them up late at night. Next slide. From there, we develop a hotspot. So we want to be looking at the right spot. The Great Lakes is huge. We only have a limited amount of capacity and resources. So we want to focus in on where it's most likely these species show up. And this is a great example of a partnership that we have with the Nature Conservancy, who does the modeling work behind this hotspot analysis. Our Great Lakes work is based on invasion risk at sites throughout the basin. TNC has developed a model using surrogates that create an additive model that describes propagular pressure. And from this, we can pick out the spots that we're going to hit throughout the year. Next slide. So I'm hearing, Nick, this is an eDNA webinar. Why haven't we talked about eDNA? And that's because it's a tool. And this is where we bring eDNA in, right? So we have a lot of species in a lot of habitats, and we have a broad spectrum surveillance. But our partners have told us that bighead and silver carp are important. And so how can we focus some of our effort with the appropriate gear? A lot of gears can't detect them, especially in some of these habitats. And this is where eDNA comes in, and that we can focus on specific areas and cover more of the basin, particularly focused on silver and bighead carp. For our program, our field offices collect and analyze over 4,000 water samples in our pursuit of early detection for these two species. Next slide. Once we have a detection, and like I said, we have, in addition to the eDNA, we have our broad spectrum surveillance, which is traditional gears. And so there's a little bit of a variation on the processes in this area of it. When you have a fish in hand, it's verifying the species. And if we don't have that institutional knowledge, contacting experts to verify that before proceeding to alert our state partners. With eDNA, that's more of verifying that what we truly have is DNA in the water. So going through our QA/QC protocols, looking at our controls, making sure everything checks out, so that at the end, we are really confident that what is actually in the water is DNA. Again, I want to stress that it's DNA. It's not an actual live fish. And that's what we'll discuss later on in the wheel. Next slide. That gets us to our communications protocols. Here's the one displayed for eDNA results. We have a separate one for a fish in hand. I can't stress enough how having communication protocols, communication SOP ahead of time helps prevent a lot of issues or barriers or misunderstandings that come up. Having that in place gives confidence to our partners and us, and as we engage stakeholders and people that are interested in AIS. Next slide. And then finally, we come to risk analysis and response. Okay, so giving that, particularly with eDNA, meeting with our partners and discussing what those results mean. If it's a single positive, looking at alternative pathways or where it was found or timing of the year, what that potentially means, and coming up with a game plan moving forward, which oftentimes includes press releases, an additional follow-up eDNA sampling, or adjusting our traditional sampling, moving up in time, or focusing it on that area as a response. As you'll notice, the unicycle wheel, I call it the unicycle, the wheel part happens on an annual basis. The top part, the horizon scans, development of watch lists, and identification of hotspots, doesn't happen every year. It happens periodically. We don't do it once and done. We come back and revisit it after a set amount of time, usually about in the 3 to 5 year period. Next slide. As I had mentioned before, we have a lot of state partners, state and federal and non-governmental partners that participate in this. And I'm excited to turn it over to one of those partners, the state of Michigan and Lucas Nathan, to talk about how their relationship with eDNA and how they collaborate with us and how they utilize it at a state level. Thank you.
All right. Thanks, Nick, and hello, everyone. As mentioned, my name is Lucas Nathan. I'm with Michigan DNR Fisheries Division. And really today I'm just providing a brief overview of some of the things we have going on in the state of Michigan using eDNA, including our partnership with Fish and Wildlife Service. Next slide. So as a brief overview, I know I saw a lot of different affiliations come through in the chat. So I know there's a lot of folks from a lot of different jurisdictions across the country. Just want to provide a brief overview of our invasive species program, knowing that it could be in some ways similar, but in other ways is different than your own. So here in Michigan, we have our collaboration is among 3 different departments. So we have agricultural and rural development, environment, Great Lakes and energy, and natural resources. All 3 of those departments come together to develop and implement a wide range of invasive species projects and efforts across the state. And we do so under the guidance of 2 different management plans. So we have our aquatic and terrestrial state management plans that identify priorities, everything from prevention type efforts to surveillance and monitoring, all the way to control and management. Also within our program, we're fortunate that we have an invasive species grant program that we issue to partners across the state and region to implement a variety of projects. And that's particularly relevant as I start talking about some of these eDNA type applications, as our program has funded eDNA type work in the past. Next slide. So just a really high level overview of some of the eDNA applications that we have going on in the state. This is certainly not an exhaustive list, but just want to give a sense of some of the recent projects that we have here in Michigan. First, and perhaps one of the most extensive one is the collaboration that you've already heard a little bit about with the U.S. Fish and Wildlife Service, focused on invasive carp surveillance across the Great Lakes, and we have several high priority locations here in Michigan that we're keeping an eye on for those species. Additionally, we've had projects focused on evaluating detection probabilities for single species like grass carp, and also doing whole fish community surveys in comparison to some of our other traditional methods of invasive species surveillance and fish community surveys that we have going on across the state. Similarly, we've done some work evaluating eDNA as a surveillance strategy for red swamp crayfish, and also used eDNA as a sort of pre-post control evaluation. Both of those projects that I've referenced have been in collaboration with partners at Michigan State University. And then also on the terrestrial side of things, we've also had a few projects over the years looking at a few different species, including feral swine surveillance and evaluating the use of that technique for detection of that species using stream-based water samples. There's also been some work going on focused on hemlock woolly adelgid surveillance using air-based filter methods. And those 2 projects were completed or are ongoing with partners at Central Michigan University and Grand Valley State University, respectively. Next slide. And what you might have seen from a kind of common theme across all those examples I just referenced is that our involvement from a state perspective is oftentimes as a project partner. It's rarely done alone, and really not at all in the case of eDNA. And this is largely due to the fact that from our perspective and in our program, we don't necessarily have the internal capacity or the lab infrastructure or ability to lead some of these projects on our own currently. So oftentimes we are very heavily dependent on some of these external partners to work with to be able to use eDNA as part of our surveillance program. So oftentimes we are sort of serving as either the content expertise or helping to identify the issue or the management need, and then providing some additional expertise on the system or species that we're focused in on. Next slide, please. And to give a kind of a more sort of detailed example of how exactly we're using eDNA to inform response efforts, I want to just sort of walk through the example I've already referenced a few times here, which is the invasive carp surveillance efforts that we do in coordination with Fish and Wildlife Service. This is one of the most extensive ones we have going on, both across the state, so spatially and also through time. So this is one that is certainly one of our most active eDNA type efforts. As you heard from Nick already, it involves a lot of coordination between our respective organizations. Usually starts in the winter months where we have planning meetings identifying priority locations of where we think carp might be most likely to show up, if at all, and helping to kind of provide some of that guidance about the local systems that our staff know very well. We can help to coordinate where those efforts should be best placed. As Nick also referenced, communication is a very big part of this partnership, and making sure we have protocols in place to, if detections do occur, how do we handle the communications internally, and also how do we develop coordinated communication externally as well. So unfortunately, we've had the opportunity to work through some of those examples here the last couple of years. We have had a few detections. And when we do get detections, usually the process then involves a heavy amount of coordination and communication between the organizations. Oftentimes we look to do follow-up surveys, either using eDNA or traditional methods. And if the former, it usually results in a request back to Fish and Wildlife Service to implement additional survey efforts if they're not already planned. And then again, the communication part is a huge part of it. So we have multiple instances where we have coordinated communication plans. We do joint press releases and things of that nature to alert all the folks across the basin that need to be aware of some of this information when it becomes available. And really, overall, eDNA, at least in our program, is viewed as sort of providing one source of information to guide our management decisions. As Nick referenced, it's one tool that we can turn to, and certainly it's a valuable one, and it's an effective one when applied in some of these methods. So it's one way that we can collect information about potential risks on the landscape and help us make better decisions of how to respond to any given risk. Next slide. One way that I've really appreciated being able to kind of walk through or communicate how to handle eDNA detections, knowing that sometimes it can be challenging to effectively communicate how we should be responding to a single detection of a species or a single replicate within a batch of samples. Having documented protocols and decision trees to support and point to help provide justification for our actions is something that I personally value. Just provide one example of such a tool that's available. This was some work that was completed by Amy Welsh and partners a few years ago and funded by the Great Lakes Fishery Commission. And I won't walk through this whole diagram, but in general, it provides some guidance on how to respond to different levels of detections depending on the perceived risk and potential other sources of DNA. And with repeated sampling, it sort of provides a good way to kind of think through a problem of how do I respond to eDNA detection. So if you're interested, I believe that these documents, as well as other supporting documents associated with this product, will be available to all the participants here. Next slide. With that, I really wanted to end on just a couple of things that tend to dominate my brain space when it comes to eDNA and using eDNA within our program. And as part of that, what I also see as some opportunities moving forward or some priorities for folks that are interested in using this technique or that already are using them, things that they could be focused on. So one of them, as I just kind of left off the last slide, is interpreting and communicating results. If you've ever had to be in a position of communicating what to do with low levels of detections of something with eDNA or eDNA results that might conflict with things we think we know about a system, it could be a challenge. So next slide. And where I see as an opportunity there is having consistent protocols. So this is both field-based, lab-based, communication-based, and response-based. As I just mentioned, there's some helpful tools out there to help kind of think through and communicate how we're responding to some of these detections. And doing so consistently, in my opinion, can help address some of that confusion that might come along with interpretation and action associated with eDNA. Next slide. So yeah, that sort of just hits on the decision support tool capacity. Again, there's plenty of them out there. I just referenced one of them. It's certainly not the only one, but it's something that can be used as a helpful process to think through and communicate these types of results. Next up, another challenge that oftentimes can be present, especially in the context of EDR type applications, is processing timeline. Currently it's not uncommon that samples would be taken and you might not get results for weeks or months later, depending on lab capacity and associated timelines to get those samples processed. So that can be a challenge when it comes to trying to respond in a rapid way. There are a number of ways this could be addressed, but one opportunity that I'm really excited about personally is opportunities for more real-time detection methods. So more stream side or system or site-based detection techniques that could be available to help provide some of that more real-time information to help guide on the ground response efforts. Next slide. And the last one I'll mention, this is ever present. This is really not an eDNA specific thing, but capacity. That's always a challenge for all of us that are involved in natural resources management, invasive species in particular. There's always more to be done, and oftentimes we don't have the time, money, or resources to get some of this work accomplished. Next slide. And as I've already kind of referenced really throughout my presentation here, where I see the opportunity there is with partnerships and external funding. We rely heavily on both of those things for really everything we do, not just eDNA, but certainly eDNA in particular, given the more technical expertise that is needed and some of the lab capacity and facilities. That's something that we'll likely continue to rely on very heavily in the future as we look to continue to use eDNA as part of our invasive species program. And with that, that's all I had, but I think we're up to a Q&A session.
Yes, thank you very much. And we have a few questions that have been rolling into the chat. So the first one is actually from Tammy Davis. And Tammy wanted to know if Michigan would be willing to share their eDNA communications protocols.
Yeah, I'd be happy to. We have a regional communication protocol that we tend to use. That's not specifically eDNA. It's really just invasive fishes generally, but I'd be happy to share that if others are interested.
Great, thank you. And the next question is from Brian Turner, and he says, seems clear what the response is when you get a positive detection and follow-ups, etc. But what about negative results? What degree of confidence is there that the lack of detection can generally be considered absence of the target species?
Yeah, Nick, did you want to take that one first, and I can add to it?
Sure, I can do that. So that's a real good question, and that a lot of that I believe has to do with that upfront planning. The number of samples you collect, where, when, and a lot of that's based on biology of the fish, and then I think an important step is occupancy modeling. So, taking samples in known locations and then modeling those so you can tailor your sampling schematic to increase that probability of detection. Again, I say it's a tool like a traditional gear. Just because you don't detect it like a traditional gear, if you don't catch it, doesn't necessarily mean it's not there. But we can put some level of confidence on that with occupancy modeling. Lucas?
I don't have much to add. You kind of stole everything I was going to say, but I would just echo that, certainly we know that any technique is not perfect. It doesn't matter if it's electrofishing, netting, eDNA, any technique for any species is not perfect. And I think exactly what Nick said, like having a better understanding of the probability of detection, given the species and system and tool that you're using, regardless of if it's eDNA or not, in my opinion, is a high priority, just to know how confident you should be in those zeros being indicative of non-presence versus a true absence.
Great, thank you. We have one more question before we hand it over to Meredith, and that question's from Paul Zajac. When deciding on hotspots or outfalls from publicly operated treatment works mapped and considered, the influent and effluent of the tertiary treatment plants, are they sampled for eDNA?
I can take that one. Yeah. So we typically view treatment plants as like a potential source of DNA that might not be from a live fish, and so we try to plan our sampling away from those where it won't be impacted by the outflow from those treatment plants.
Thanks. Okay. I think we're going to hand it over to Meredith, and there will be some more opportunities for more questions. So thank you, Lucas and Nick. Really appreciate that.
Yeah, thank you. So on to our next presenter, Meredith Bartron. Meredith is the U.S. Fish and Wildlife Service Center Director of the Lamar Fish Technology Center in Lamar, Pennsylvania. Meredith's previous research focus was application of genetics to conservation issues, including research and application of environmental DNA. Meredith is part of the EDRR, the National EDRR Framework, as co-lead for the market development project and interim coordinator of the molecular lab network, which she's going to tell us about. Take it away, Meredith.
Thanks, Kaylin. And hopefully you can hear me okay. So thanks so much for joining. I'm going to kind of expand a little bit of what the system that Nick and Lucas were talking about in the Great Lakes to a molecular lab network that's being developed to serve to work across the country. So I'm going to jump right into that. So next slide. So going to provide a little bit of context for why we're developing a molecular lab network. There was a coordinated national EDR framework in the US has been recognized as a critical need in multiple strategic planning documents that have been developed over the last decade or so, as well as in a special issue of biological invasions, and that was published in 2020, which highlighted 12 different papers that related to a need and options for this blueprint for a national program that would provide early detection and rapid response for invasive species. These efforts have formed the blueprint to facilitate the communication, collaboration, and innovations necessary for an effective national EDRR program, especially for non-agricultural invasive species where such a framework was not comprehensively established on a national basis. So recent funding from the bipartisan infrastructure law has established or has enabled DOI to advance many aspects of the kind of as this architecture framework to enhance EDRR capabilities and to coordinate that on a national level. And really with the goal to advance transformational outcomes that might not otherwise be achieved without that support. So that bill funding has really has enabled DOI to make these advances. And DOI itself is committed to advancing that framework in collaboration with other federal agencies, states, tribes, territories, local governments, and other partners in invasive species management forums. So this is really meant to be a collaboration initiated and funded through DOI, but bringing in various entities and perspectives to make this framework a success. So this here is just the working mission statement right now for the EDR National Framework, which really speaks to that strengthening coordination and capacity, one of which is the molecular lab network. So next slide. So this kind of unicycle that Nick mentioned, kind of the structure of everything from horizon scans and to leading to analysis and identification of invasive species, that workflow, we kind of distilled it down into these 4 components of this plan, detect, report, and respond within the framework. And there are many projects that have been identified and established and initiated within the framework to get to different aspects, to address different aspects of that plan, detect, report, and respond framework. I'm going to focus a little bit more on the molecular aspect, the molecular projects that have been funded. And 3 projects are within the plan phase. Those include the eDNA Toolbox, which is an informational resource to learn more about eDNA. Marker development project, which will develop and validate markers for eDNA applications, and the genetic material repository and network, which provides DNA and sources tissue for that marker development and testing. Within the detect component of this workflow, there are 3 more genetic related projects. One is where they're actually applying those genetic tools for invasive species detection. So that's the ReadyNet autonomous sampling project for sample collection, the point of use project, which is an in-field application of eDNA, and then the molecular lab network, which I'll be talking about in more detail. So next slide. So the molecular lab network, kind of our mission statement or our operational kind of definition is really to be a network of genetic labs that are dedicated to providing consistent technical support to facilitate the implementation of early detection and rapid response monitoring. So we're meant to be so this bill funding is being used to support the development of these labs to form this network. The targets for the MLN will be both aquatic and non-aquatic species detection, with an emphasis on aquatic species for at least the first year, just based on kind of where the science is for this broad scale AIS detection. We're really meant, as I mentioned, kind of talking about some of those other molecular projects funded through this EDRR framework, is to really leverage those existing efforts, both within the framework and outside of the framework, and collaborate with other EDRR framework projects to advance both the science and the application of eDNA for early detection. The molecular lab network clients are projects, at least right now, are projects within that EDRR framework. So there are two groups within the EDRR framework of surveillance projects. One are these invasion hotspot pilot projects, of which there are 6. And then there's asset-based protection projects that also have some component of molecular tools or detection for invasive species screening. So the goal of the molecular lab network is really to be very engaged with those clients. So engaging with the folks that are doing the sample collections, everywhere from before samples are collected to understand what the needs are, the species being addressed, and ensuring that molecular tools are appropriate for that species, for that surveillance and monitoring efforts, all the way to post-collection, where we're sharing the results and assisting with any interpretation to make sure that there's an understanding of what the results mean, as well as what they might not mean. And then since this framework and the lab network is being built specifically for these projects, they are being equipped and developed and staffed to be able to provide timely turnaround and rapid response, with the goal being that early detection, really making sure that we are able to meet those time requirements to get that, especially if further action might be needed. Next slide. So within the EDRR framework, or within the molecular lab network, there are 2 components. One are surveillance labs, and the second component is this implementation science support lab. And when we were developing this lab network concept, we recognized that the field of environmental DNA is rapidly evolving. There are new tools, there are new techniques, there are new methods that are being developed and identified kind of, it seems, almost daily. And so that ongoing science support need is really critical to enable the surveillance labs to make sure that we're using kind of the best available methods, that they're being implemented in the right way, and really to ensure that the science that's being used and interpreted is being done in the most proper way, back to the clients, right? So within the surveillance labs, there are going to be 3 surveillance labs, and I'll show on a map in the next slide. But right now, the goal of that surveillance lab is really to be that interface with the folks on the ground doing the surveillance. They'll be communicating with the partners, they'll be providing the sampling protocols, and on all that sample design and guidance. They'll be really engaged with the partners to make sure that eDNA is being applied in the way that is as intended by the folks that are needing that type of information. In the lab, they'll be doing all the lab analysis, managing the data, interpreting the results, and then coordinating with that implementation science support lab group to identify if there are new species that are being identified of surveillance need, new sampling methods that might be needed, or any sort of kind of the research need, both from the application side for eDNA as well as implementation across multiple labs or within those labs that are part of the network. For the next, if you can click it again so we can see the box. There we go, thanks. For the implementation science support lab, they're going to be working to address those science needs. And again, the science needs are not just the implementation of eDNA, but also implementation of eDNA across multiple labs, to make sure that that work and that science is being done consistently. So They'll be working and coordinating with some of the other framework projects to facilitate tech transfer. So like the marker development team, making sure that we're working with them appropriately and getting, more markers as species and needs arise. Optimizing the application of eDNA, and then also coordinating with the surveillance lab. So these two components are really meant to be very intertwined to make sure that we can successfully implement a large-scale eDNA surveillance program. Next slide. So who is the molecular lab network and where are they going to be? So as I mentioned, there are those kind of two branches. So the surveillance labs, there are 3 surveillance labs. One is going to be within the Fish and Wildlife Service at the Alaska Conservation Genetics Lab up in Anchorage. The second is at the Fish and Wildlife Service Southwestern Native Aquatic Resources and Recovery Center. That's in Dexter, New Mexico. And then the third surveillance lab is the Fish and Wildlife Service Southeast Conservation Genetics Lab that's located at Auburn. Now the Implementation Science Support Lab is located at the USGS Forest Rangeland Ecosystem Science Center in Boise, Idaho. And then all 4 labs are going to be coordinated by a molecular lab network coordinator as well as a data analyst. So those 2 positions will serve all 4 labs. And it's really meant to be, this is a collection of a coordinated effort among all 4 labs to make up the lab network itself. Next slide. So to make this happen, there's kind of 2 groups that need to be 2 topics that need to be addressed. One is the infrastructure. So the intent is that all of the labs are going to have the same equipment. So we have standardized operations. The workflows, especially really with this particular focus to minimize contamination, are meant to be standardized across all 3 of the surveillance labs. That the staffing is being hired that are specifically to work on these molecular lab network projects for those surveillance projects, so that way we can make sure that analysis is done in a timely manner, and that there are sample and data tracking systems that are being developed, so that way we can look in real time to see what the sample data is, where the status of those samples are, and then also what those results are and how those are being managed. So that operation or infrastructure wise, there's quite a bit that needs to get done. Operations is this on the same way. So just as much as we want to have standard equipment, we want to have standardized protocols. So that way each lab is processing samples in the same way so that results are consistent regardless of which lab you're working with. That operations is really critical to make sure that we have staff that are operating the same way. So we'll be implementing proficiency testing as well as a QA/QC program to guide operations. Just as much as interaction in like the lab operations are, we want to make sure that we have streamlined communications so that way folks know how to interact with the MLN and also the information flowing into and out of the molecular lab network is consistent. And then that way we're also working to identify those research needs and that those are being communicated among all parts of the MLN. Next slide. To make all of that operational standpoint work successfully, we're right now in the phase of developing all sorts of protocols to guide everything from some staff operations to all the lab operations and all of the parts in between from sample collection to communication of results. So the intent is that all of these protocols will be followed by all of the labs that are part of the network. That these protocols are peer reviewed and consensus based, so that way all of the labs have bought in and we've gotten external viewpoints to make sure that we're using the best available science and the best available protocols being implemented within the lab. We also realize that given that this is a rapidly evolving science and field, we want to make sure that those protocols are being reviewed annually, so that way we are adapting over time to make sure that not just we're using the best science, but we're also using the best science across multiple labs. Next slide. So another question that we might be getting is kind of, okay, so we have this lab network. What are we basing our species that we're doing our analyses on? So I wanted to provide a little bit of background for that. So going back to that unicycle, kind of the early phase, and I think Nick touched on this in terms of like horizon scans and watch lists, hotspot analyses, partner input. all of these multiple sources of information are going into developing kind of what species are needed for to have molecular tools available for detection. Within the framework itself, there are 3 projects that have been funded that are working to generate some of these lists that are helping to feed into some of this, the potential lists of species, as well as the different surveillance projects and the that are being stood up as part of the framework. So specifically within the framework, it's the Horizon Scan Watch List project, the Hotspot Analysis project, and Inhabit are helping to identify species that are of concern. That much longer list is then being fed into the marker development group. And also, there's a evaluation process in terms of what species would be good for or viable for environmental DNA surveillance. And then the marker development group, which I mentioned earlier here, and here is also going to be working on then taking those lists of species and identifying and either developing or validating existing markers that can then be used for the molecular lab network. So that way all the labs are not only using consistent protocols, but we're using the same markers to detect the species that are identified as of need by partners. Next slide. And just a little bit about what kind of tools the molecular lab networks is going to use. So there's multiple methods of eDNA analysis that are available right now. And the 2 primary ones, however, are quantitative PCR and metabarcoding. Both of these tools have pros and cons, and for a variety of reasons, each of the different tools are certainly viable for eDNA analysis and especially large-scale projects. Initially, the molecular lab network is going to focus on applications of quantitative PCR. This is in part because we're working closely with the marker development team to get marker-specific qPCR markers developed for those lists of species that are being generated by those other groups that I mentioned. Metabarcoding is something that we also want to apply. I think some of the challenges, especially for large-scale projects, is that metabarcoding also, as part of the application, requires a comprehensive reference database, which isn't necessarily available at the scale that that some of the sampling is going to occur. So that's something that is being developed, and additional work is needed to ensure that we could use metabarcoding on a broader scale. Some geographies, however, this might be more that those sort of reference databases might be more readily available, especially depending on taxa as well. So at least initially, the ML1 is going to focus on quantitative PCR with the goal of moving into metabarcoding over time. Next slide. Thanks. So how is one of the questions is how would groups interact with the molecular lab network? And as I said initially, our client group, that's all right, Carrie. Initially, our client base really are these early detection rapid response framework surveillance projects. So there's these asset-based protection projects, which are within the different bureaus of Department of Interior, and then these invasion hotspot pilots, of which there are 6. And that's where these pilot projects are kind of distributed throughout the U.S. The first pilot that's been stood up is in the Southeast, and that's gotten really well established and is being coordinated by Wes and James Ballard. And these other there are of the 5 other pilots, those are being stood up, and I guess, in my understanding, is the 2nd year. And so some of those are pretty well formed, and some are still in those kind of that pilot forming stages right now. But that's an opportunity for additional partners outside of DOI to be engaged, because the pilots, as my understanding, at least in the Southeast, is working really closely with states and other partners as well. So in terms of how these groups then will work with the molecular lab network, or how will those interactions go? So the pilot projects or the surveillance projects, as they identify eDNA surveillance as a tool that they'd like to engage with, they'll be reaching out to the molecular lab network and providing information such as the species that they want to detect, or the number of samples that they plan to collect, and kind of some of the sampling parameters in terms of the habitat they're sampling, if it's, lakes or streams, what kind of season they're going to be sampling based on, which is information that is needed by the molecular lab network. So they'll reach out to the molecular lab network, who will then click again, provide guidance to the partners related to eDNA sampling design to make sure that it'll meet the eDNA collection protocols, and the design of the project will meet the needs of the surveillance effort itself. They'll work on providing all the training for sample collection, as well as all the sampling protocols. In most cases, they'll be providing the eDNA sampling supplies, so the filters to actually filter all the water samples for, at least initially, for the aquatic-based projects. They'll be doing all the analysis and then getting those results back to the partners. So it'll be a really close connection and a flow of information between these two groups. Next slide. So to make this happen, there's a lot of steps that need to occur, and kind of the tasks at the top in green are all kind of related to that lab preparation. And many of those tasks, they've all been initiated and we're at various stages of completion. Most of the labs have been staffed, and the equipment is in place. Right now, we're working on with another project to develop the sample tracking and lab management software to ensure that we can follow samples, and all the information is attached with those samples can be tracked, and all the lab operations and processing of those samples can be tracked. All the protocols that I mentioned and all the QA/QC and communication plans, those all need to be, especially the communication plans need to be developed, and especially that needs to be developed with partners. And so, we're not quite there, almost. So we can make sure that we're working with partners to develop and understand what those communication needs are. Right now, we're waiting for kind of some of those planning lab preparation steps to be done, and then also for markers to be available. As soon as those markers are available, assuming that we've got all the other kind of lab prep tasks checked off, then the labs will be able to be open and able to receive samples. So we're hoping for that to be soon. Next slide. So kind of what will be available, what are the products of the molecular lab network? All of the field and lab protocols, all the training materials, and all the validated qPCR markers that are developed through that molecular marker development project. We're all going to be available, and those will be available on the invasivespecies.gov, the SIREN website, which is meant to be kind of a housing of a lot of the information that's being developed through the EDRR framework. This has been kind of given a soft launch through the SIREN project a couple weeks ago at the NIASMA conference. And this is a great resource. Amy Agnew is the lead on the project and has developed this incredible resource that SIREN is going to be built out even more too over time. And this is where we hope to house a lot of the molecular lab network information that would be available. So that will be a product. The products from the MLN will be available on the SIREN website. And then also all the genetic eDNA monitoring results will also be a product of the MLN. And we'll be working with partners to understand where best to house those. And I think that's it. Last slide, I think, is, just questions. So thank you so much for your time. And I'm happy to answer any questions.
All right, thank you very much, Meredith. Really appreciate that. So there were a few questions that did come into the chat. The first one was from Lisa. And Lisa wanted to know, are there any plans for the MLN accreditation for state, tribal, and NGO eDNA AIS labs?
That's a great question. So as we're kind of building up the surveillance labs and implementation science support lab to follow kind of this standard set of protocols, we've talked about if there's an accreditation process, at least internally within our lab network right now that's being built, what that would look like. Over time, and certainly we want to make sure that those protocols are available for anyone to use or as a resource, just to understand kind of at least what the lab network itself is using. We haven't necessarily yet set up a system to incorporate more labs into that lab network, as we're still trying to get started, and the funding is kind of specific to this group of labs right now. But certainly being able to share those protocols and the processes that we're using and develop a broader community would be great to do. I'm not sure what that accreditation plan would look like, but I think that's something that would be great to have a conversation about. And another great resource too, I think that's also addressing some of that or having some of those conversations is the Gedwig group. And I know we've got some folks that are engaged with Gedwig on the line. Katie Klimas, who right now is one of the leads for Gedwig, is on, and that might be another option too.
Great, thank you. And there was also a question from Paul Zajac relative to metabarcoding. Cyprinids are a very large family. Can you discern a carp species of interest using metabarcoding?
That's a great question. It depends on which species. I think there are some overlaps in some of the sequence for current metabarcoding practices. And I think that's where understanding what the target species of interest for AIS detection, as well as what also are some of the native species that could be present, to understand where, say, metabarcoding would be appropriate and would just differentiate between those suite of species versus a targeted qPCR marker. So that's a great question, and really I think some of those questions are kind of need to be addressed in terms of matching the marker or the tool with the species differentiation.
Awesome, thank you. And we have a little more time. Does anybody have any questions also for Nick or Lucas, just in general? Everybody can now unmute themselves. So Monica just delivered. So Monica McGarity says, is there a means by which marker development for a given species could be requested to eventually be a resource available to natural resource managers via Siren? Or is marker development, species development need to be driven by the needs of the DOI projects?
I'll answer that in a couple of different ways. So I think one of the goals of the marker development project is to have all of the markers that are developed and validated through their available on Siren. So that will be a resource. In terms of identifying species to develop markers for or validate through the project for use in the MLN, a lot of those initial species lists are coming from some of the pilot projects, but a lot of those pilot projects, especially this next round, as we have more pilot projects coming on board, are a lot of those are partnering with various states and tribes, and so there's opportunities to get those species on those lists that way. I know also because they're also taking into account horizon scans and watch lists and some of the hotspot analyses. That becomes a very broad list of species being targeted that could potentially be targeted. So I would say definitely once those pilots or as those other pilots are getting formed, looking for opportunities to potentially engage with those. And I know I think Kaylin might have some more information too on what those pilots are, additional pilots are that she could share. And I see Hillary's got her hand up, so maybe she can weigh in as well.
Hillary, you can go first.
Sure. Sorry about that. Hey, everybody. Great talks. Thank you so much. So just one more thing to add to what Meredith shared about the opportunity to help identify markers. I think that was an excellent question. And as Meredith said, initially we are focused on the kind of getting this pilot approach up and running in general. And so it is focused on a narrower set of projects. As Meredith said, the asset-based protection, which is focused on DOI managed lands and waters, and then those 6 regional pilots. And so as Meredith indicated, there's one in the Southeast region, there's one in the Great Lakes Basin, inland lakes and rivers that Nick and Amy McGovern are helping to facilitate, the North Central US, in collaboration with the Blackfeet Nation and potentially other areas as well. The South Central Alaska and the West, which is going to be focused on terrestrial species, and then the US territories, and that's still being scoped. So each of those 6 regional pilots have a bureau lead that is working in collaboration with their state and other partners to help scope what the interests are in those areas. And then looking beyond Bill, what we've, as Meredith mentioned in her presentation, the view is for the National EDR framework to be interagency in nature. And so we do hope to set up some kind of structure so that we can have a national conversation about what the highest priority needs are for marker development. So again, we're kind of piloting these collaborations now, but in the long term, we absolutely do see setting up a mechanism that there are federal, non-federal communications about what the greatest needs are. And we did, this is maybe getting a little too detail oriented, but we did ask the National Invasive Species Advisory Committee, which is a non-federal group, and the Aquatic Nuisance Species Task Force. They formed a subcommittee. And one of the questions we asked them was, what kind of a formal structure can we set up going forward so that we can have these kinds of conversations and identify the most pressing needs in a more formal way? So more to come on that. And again, appreciate that question. I'll turn it over to Kaylin if you wanted to add anything further that I might have missed.
I'm good. And I see there's one more question in the chat.
Yeah, we'll take one more question and then we're going to jump into breakout rooms. So thank you for that explanation, Hillary. So Paul has another question. When a marker from Maine located European green crab was used in Washington state, and positive returns were noted for 3 endemic crabs as well as European green crab, do you consider location when developing a marker?
Yeah, and I think Steve probably is up to do one of the next talks should be from the marker development team project. Yeah, it seems like that would be yeah, location and both for where markers are developed and where markers are being applied is part of that validation process that the marker development team is engaging is going to employ. So it seems like yeah, those are really critical because some species have very different sequences throughout the range, and that could impact the marker being used. So I don't want to get too far in the weeds to steal Steve's thunder, but I think that definitely is going to be addressed within the marker team.
All right, thank you so much. So really appreciate our 3 presenters and all the information that they provided. And now it's your turn. So we're going to jump into some breakout rooms. There will be 5 breakout rooms, and each room will have a facilitator and a note taker. And we have a set of questions that we'll be asking you. I'm going to post them in the chat really quick, but you'll also have access to them with your facilitators and note takers, and you'll have about 20 minutes to discuss these things. So I'm going to open all the rooms. Everybody can jump into the rooms. If you need help moving into a room, just let me know. We'll have you guys head over and chat about some of these questions based on today's presentations. All right, well, welcome back, everyone. There never is enough time for breakout rooms. I was jumping around a bit, and I guess 2 key points that I heard as I jumped around were marker development is something that folks are really interested in, and communications. So we have about 5 minutes left, and we do have the notes in those notes documents. So I will be working to pull all those together and summarize and all of that. But within the last few minutes of today, since we won't have time for all the groups to report out, were there some big things that you think would be good for the group to know that came up in your different groups in your discussion? Susan?
Hey, so one of the things that came up was our folks were really excited about standardized protocols, whether it was for the field sampling or the laboratory, but there was also emphasis on these protocols being validated, something that if they get challenged in court, they can stand up and say, yes, these are good standards and we're all using these same standards. Or if they need to send a sample to an additional lab or another lab to verify samples, they're using the same samples and they're assured things are good.
Great, thank you. Jenny.
Just since Susan brought up standards, there was some conversation about guidance and standards for interpretation and consistency amongst labs as well in our group. So just to extend that standards, including field, lab, and then also sort of that decision and outcome basis in terms of how the data is used.
Yeah, definitely. Amy.
In addition to the communications and sort of the jurisdictional authority, decision making as a real prominent piece there, there was also something that was brought up that I thought it was interesting that, what about the sort of opportunistic sampling that's happening with groups across the basin? How are they tracking the work that's currently going on and knowing kind of who's doing what where? And also the opportunistic sampling efforts that are happening, ensuring that communication protocol can be shared and perpetuated by those types of other groups that are also doing eDNA work was a concern.
Great point. Thank you. Nick?
Get back to the standardization. In my group, there was agreement that standardization is good, but there was concern about the level of standardization, the detail, if it was down to the, for example, specific pipette that you need to use and how that impacts, potentially other private labs or other labs associated with other groups, what that means for them, if that almost excludes them. And they noted that it's particularly important. I had Western states and territories, and there's not, proximity to labs is important, and there's not anything close right now for them.
Yeah, that's a good point. I definitely know that territories do struggle with a lot of those resources as well. Great. Are there other groups that also had some key points that they wanted to share? Or just any, aha, cool moments that you learned about today that you just want to let folks know about? And Teresa Tom put in the chat, we ran out of time in our group, but one thing that could be helpful with the molecular lab network would be helping streamline sample processing to increase timeliness of sample results. For example, some labs may be overwhelmed with samples given an emergency response to AIS, and hopefully other labs in that network may be able to handle that increased workload. So much they're sharing there.
Also, Carrie, to add, I think that there are a lot of really good efforts ongoing already in some different regions, and hearing about zebra quagga work in the Pacific Northwest, invasive carp work, crayfish work. Is there a way to kind of connect us in a way that we're using some of those successful and ongoing programs as a springboard to improvement and sharing across nationally?
Yeah, that's a great point. Anybody else? We tapped you out, right? Well, I want to again thank all of our speakers and also all the folks behind the scenes who have been helping with this webinar. It takes a lot to make these happen, and really want to throw out hands to Kaylin for all her coordination efforts on today. So she has really kept the whole project running. So I appreciate all of your time. And again, I'm going to synthesize what has come out of the notes. If there are other things that crop up in your mind as you're sitting down, eating a snack afterwards, please feel free to reach out to us, and then we can add it to some of those documents. And keep an eye out. You know, we're going to be using this information to help with some of the other future webinars in this series. So we're really listening to you all and kind of your needs and your questions, and trying to respond with these different resources and webinars. So thank you again, and we'll be sending out the recording and all the notes within probably 2 weeks. And but if you have any questions in advance, feel free to reach out.